Good afternoon, ladies and gentlemen. I warmly welcome you all to the fourth and the last session of the second day of the 13th International Research Conference of General Sir John Kotelawala Defence University. Now, you are hearing us in the basic and applied sciences section based on the theme of making growth and security viable.
This session will be judged by esteemed panel of judges. This session will be chaired by Dr. S.H.N.P. Gunavikrama from General Sir John Kotelawala Defence University. We cordially invite Dr. S.H.N.P. Gunavikrama to commence the session.
Thank you. We have reached the final technical session of the basic and applied sciences stream of Kotelawala Defence University International Research Conference in 2020. And in this session, I would like to declare that we have three very important in-depth analyses in three different areas. One study represents computational biology, while other two represent molecular biology and molecular genetic areas. Even though they seem like pretty similar, they represent actually diverse areas of modern molecular biological approaches. So this is where we are heading in life sciences now. Things are developing fast based on molecular approaches, and in that context I would like to say that these three studies, and in fact the technical session 4, which we are undertaking right now, seems to be very, very important and refreshing. The study 1 is each study is given a 20-minute time period for presentation, which will be followed by a 5-minute Q&A, and that is the session format. And due to COVID-related measures of government of Sri Lanka, we have had to conduct the session according to the rules and regulations prevailing in the country in that context. Therefore, we resorted into online presentations and online system where physical presence of the authors and the audience is not really necessary. So I would like to invite the first speaker of the session in this context to present the study titled Effect of HTAC Inhibitors on Stability and HTLP Enzyme and In Silico Approach. It's a computational biology study. The study is presented by Mr. R. Dushnan and the study is a collaboration between the Open University of Sri Lanka and the University of Colombo in Sri Lanka. I would like to invite Mr. Dushnan to make his presentation. Mr. Dushnan, are you there? Yes. Yes. Yeah. Mr. Dushnan, please listen. Mr. Dushnan.
Actually, it's difficult to predict an overall definition of what constitutes cancer. However, simply it can say that uncontrolled multiplication is to produce the human that can interfere with the function of the organ. Cancer can cause by two major reasons. They are genetic causes and epigenetic causes. Genetic causes are irreversible modification, but epigenetic is reversible modification. Therefore, epigenetic modification can kill by the drugs. So, this work is talking about epigenetic modifications. And mechanism, transcriptional selective mechanism of histone receptase enzyme in histone. Histone receptase enzyme induces positive charge of the lysine receiving in histone. Therefore, therefore, minus charge positive move from DNA is affected by the histone. So, the increase the affinity between histone and DNA will increase. So, transcription factor regulator may be inexcessive. Therefore, transcription silencing is take place. So, abdominal termination is taken, what is called as cancer. And the objective of my project is develop a computational method to investigate techniques or molecular description of relative and check off in bigger context. Histone may exercise inside. When externally traditionally related the use of cancers in clinical trials which reduce the length of the time and cost of studying cancer research. And the result, there are two sections in my methodology. One is investigation and another one is analysis. Investigation. Protein optimizes the laboratory volumes by the AC processing. The processing parameters are generated processing product for its organic server. Now the complex of placed at the center of the simulation. The box size is mentioned here and the box is being bottom choose. The required amount of energy for science are added to the box to maintain a neutral system. The aim of the system of minimum using 1500 steps and entire system was calibrated. After equation, 100 second energy simulation was carried out. And second fiction is reciprocity. To estimate the amount of selected HDP including complexes, the reciprocity method was employed using the RNA, amino acid, and DHO tools in only 4.6.5 platform. And these are the three standard indicators. Sapa is made 99 and 28. Sapa is the momentum drug, therefore it's considered as a reference drug, and is made 99 and 28.781 are in currently clinical trials. And results and questions. Moving several innovation. The last A, B, and C are indicated. All the systems at a training state, and the HDP enzyme in each system is shown. Controlling is in the distribution information factor. The results indicate that all the information line when with the line side of the HDP enzyme maintain a basic understanding of the complex. Also, the stability of the complex which indicated by green, red, orange are more stable than the tree like HDP enzyme. And basic variation, the configuration changes and compactness of HDP enzyme after binding of all the inhibitors to remain similar when RNA is seen. Comparatively, the HDP ETA 24781 complex shows less and constant fluctuation than other. Since the HDP Sama complex shows less RNA than the delta HDP. The results indicated on the complex system maintenance is not standard in the ecosystem. So RNA and levels of fluctuation both the same one. The systems are maintain the reasonable stability in the ecosystem. And hydrogen analysis. The hydrogen of CP is stably enzyme and inhibitors play a significant role in the stability of the system. The number of hydrogen between the intermediate and the active site of amino acids will show the affinity between the active site of the system. The number of high-level present in SPM. Some is 4, KCI 24781 is 5, and SPM 9. So, KCI 24781 contains no number of hydrogen protein, nucleic and active site. And these are the average distance, average values for the distance between two atoms because the form hydrogen, as mentioned, integer, known, and except the overall hydrogen, except the distance. The values are given in this table. Finally, in conclusion, the results of this in silico study demonstrated all the SPAK integrated complexes show more stability than the frame and SPAK enzyme. Among the three inhibitors, the results of spent by Muslim clearly show that new inhibitor BCA24781 is more effective or safe effective than but the difference in some. Therefore, it's evident that BCA24781 has potential to be used as an alternative to the reconstruct SAMA for inhibition of histone retardation in the medical field. In contrast, ASP939 shows some limitations, means it has little larger origins than others, and number of hydrogen was less. Such variations from BCA24781 and SAMA, which make ASP939 very less efficient. And maybe these are the reference, some of my references used for this study. And acknowledgement. National and Foundation is state request for this funds provided for this study. Thank you.
Thank you, Mr. Krishnan, for your nice presentation. Now the presentation is open for discussion. Yeah, I would like to add some comments on this. This is an in silico study, essentially a computational biology effort. And this study too associates the limitation of not venturing into experimental area. Computational sciences are limited to computational studying, yes, but when it comes to biology, you have to have some experimental data to prove or disprove what we have seen in your in silico analysis. So therefore, right now, a study that stops within the computer room is really highly predictive and theoretical. So I hope authors would understand this and add necessary experimental data into the key outcomes of the study, so that your conclusions can be made with lots of confidence.
Sure, thank you.
Mr. Krishnan, are you there?
Yes, I'm here.
Okay, thank you.
Thank you.
Yeah, we wish all the best for your future research endeavors, and from now onwards, we switch to the next study. Thank you, Mr. Krishnan.
Okay, thank you, sir.
Next study is from molecular biology area, and it overlaps with some parasitology as well. The study titled Identification of Wolbachia, like endosymbiote DNA, Setaria digitata genome, and phylogenetic analysis in filarial nematodes. The study will be presented by Kotelawala, M.S.A. Kotelawala, and it's a research outcome of University of Colombo. Mr. Kotelawala, M.S.A. Kotelawala, you can comment here. Of course, sorry, we start playing the recording of your study.
This is finance for genome and this is of statistical logic. have outlined my presentation in introduction, research focuses, impact of the research, methodology, results, and the conclusion. First, let's see what is Kotelawala data. Kotelawala data is a bilateral group that resides in peripheral categories in both animals. They cause leukospinal hemolysis, allegedly paralysis, and death from supports such as sheep and economic losses in animal husbandry in Southeast Asia. However, infections in Kotelawala and natural roots such as cattle and buffaloes does not cause in serious conditions. Kotelawala catches variations in lifestyle modes and inflammation. Therefore, there is a considerable need for the development of new intervention to disease control and eradication of this very parasite. Most of the viral nematodes are mutually associated with bulbage, and without bulbage, these viral nematodes cannot survive since the whole genome of bulbage contains genes required for the biosynthesis of heme, pipeline, fairy, mortar, and nucleotides. However, studies have found that Kotelawala data does not harbor bulbage mechanism, but it does undergo biosynthetic pathways of these nematodes. There are two possible explanations for this new reference line of Kotelawala data. This can be due to either present or gene terms of bulbage in Kotelawala data genome, or Kotelawala data may have developed their own sequence to code for these metabolites. So one of the main objectives of this research was to identify the levels of versatile gene translate and bulbage and Kotelawala data. Moving on to the research focuses. There is a written region Kotelawala data genome and its host-presence relationship. This research was focused on generation of the genome of Kotelawala data, functionalization and medical pathway reconstruction, identification of bulbage-like similarity in Kotelawala data genome, and finally, biogenesis of nematodes. This research will have an impact on development of new methods to disease control and eradication of this parasite, as this is the first time genomic analysis was done with Kotelawala data. And also, this research will have a huge impact on the development of novel drugs for hemophilia since Kotelawala data is considered as a model organism for hemophilia cells. Due to their close similar to germinal. Moving on to methodology. First, we collect whole samples of Kotelawala data from the peripheral retic canal from the western province. DNA was extracted using Gaussian DNA method, and the combination of extracted Kotelawala genome DNA was done by carrying out PCRs using Kotelawala data. Here are the specific findings. After that, we sample to check on the extracted DNA, and we send the secondary sample to microscopic components of sequence. Then we obtain the raw data and we did pre-processing and then assemble the genome to function as simplified techniques. Using these techniques, document analysis was carried out. In order to identify bulbage-like mechanism and DNA in Kotelawala genome, techniques were blast against several bulbage-specific reference sequences such as bulbage emergence genes using first-of-analysis program. For phylogenetic analysis, bulbage proximal sequence was blasted against Kotelawala data for Kotelawala, which are identified Oxica bulbus and bulbage environment 40 sequences, however, in severe computed database. Results of testing for each organism was recorded and aligned region was okay. Those aligned sequences of each organism were used to generate biological tree using genes and software. Firstly, alignment was done using observer method, and the biological tree was generated using the DNA joining method. Lastly, blastable basic software was used for functional relations and metabolic pathway reconstructions. Moving to the results. For screening of bulbage-like sequences, homogeneous sequences were found only for oxide and NP sequence. Sequence identity was higher than 50% and even was smaller than 4, and therefore it can be considered that sequence similarity is countable. This result provided the bioinformatic evidence for the presence of bulbage-like DNA sequences in the Kotelawala data genome. In order to prove these bioinformatic findings, Result tree is a simple estimate and it is unlikely to represent the true evolution tree with these organs. Anyway, as you can see here, Usuria, Bentley and Rubia organisms K divided second orders and settled each other from a separate K. All these mutants are all coming from a K that is related to Kotelawala, a one-picture K. According to the evolutionary systems between taxa, settled generates most and homosexual orders are finite number that contain evolution in the same. It was expected to be closely related to the lower since lower also evolutionary manifold, just like settled generated. Based on this result, we can say that settled generated evolutionary related to finite numbers with evolutionary. Last analysis was able to identify 6,055 mutation and 111 enzymes more identified metabolic pathways. According to the last function analysis, the majority of the cytogenic sequences have a higher sequence similarity to which are contained finite numbers than which are finite numbers. Here, K-analysis identified 16 enzyme protein in one nucleotide biosynthesis and one enzyme nucleotide biosynthesis. The genes encoding for methionine, glutathione, and heme were not found in cytogenic. Here the enzymes we identified are shown in color. This suggests that cytogenic are needed to do its own sequence of those biosynthetic pathways. For the better understanding of the metabolic processes, transcriptomic analysis is currently at the moment. Controls term, based on the processes, biogenetic analysis, and fossil-based sequence similarity analysis, it can be concluded that as 90% of the biogenetic contain one bacteria, and systems of similar gene data may have polarized to bacteria distant past, and restricted gene transfer may have one bacteria being into the genome while they are missing biogenesis. These are some of my references, and I would like to thank my supervisors, Dr. Kotelawala, and Dr. Chandrasekhar for their support and guidance, and Dr. Kasman from UCSC for providing resources, and Dr. Prashant from India for helping us with analysis. Thank you.
Thank you, Mr. Kotelawala. The presentation is open for discussion now. Mr. Kotelawala, could you please describe what Wolbachia is for the audience?
So Wolbachia is an endosymbiotic bacteria which is found in all of the arthropods families and in one nematode family, the Phylogeric nematode family.
Yeah.
In arthropods it is a parasite, but in nematode it has some mutual endosymbiotic relationship with the nematode. Normally nematodes does not code for nucleotides, riboflavins, E and FAE. The Wolbachia is the one who codes for those metabolites in nematodes. But in case of cytogenic data, we don't find Wolbachia endosymbiotic in that nematode.
Yeah, you conclude that horizontal gene transfer may upload Wolbachia DNA use digitata nuclear genome prior to endosymbiont loss.
Yes.
Yes, actually, your study, the data analysis in your study does not really support such a bold conclusion. You don't generate any data related to horizontal gene transfer and any endosymbiosis between the Wolbachia and this digitata in the history of their evolution. So please be cautious about conclusion making, and it has to be based purely on the data you raised. clarification on this regard, Mr. Kotelawala?
Not sure.
Are you there?
Yes, sir. I'm there. So, actually, we can't say exactly that there was a horizontal gene transfer, but we can suspect that there was a gene transfer based on the bioinformatic analysis.
Yeah, I understand. So, when you make conclusions, be clear about uncertainty in such regards. If you have an uncertainty, yes, don't be that bold, please. So, I think it's a fine study and very good effort in molecular pathology area and perhaps molecular parasitology area as well. And there's a research project going on at University of Colombo, and it's a collaboration between University of Colombo, University of California, and University and Department of Biotechnology and Bioinformatics. Bioinformatics, Bira Institute of Scientific Research in India. So it's an international effort. We have to be very commendable in this regard as well, because research, scientific research, cannot be done successfully on your own. Teamwork and collaborations are very essential. So this study clearly shows that our scientists have reached offshores in order to seek credible, dependable collaborations where they need. So it's a very good effort, Mr. Kotelawala. Thank you very much.
Thank you, sir.
We move on to next third and final study. It's again a research collaboration between University of Colombo and California State Polytechnic University in USA. And the study is authored by C. De Silva, MLD Taxtila, E. Scordato, and Samba Seniratna. Dr. Samba Seniratna is a member of the University of Colombo Academia. The study is titled Polymorphism of Dopamine Receptor D4, gene in hill swallows. Hill swallows are kind of birds from mountain human settlements. I invite C. De Silva to make his presentation. I think we are going to play the record.
Good morning, everyone. I'm Shamaka De Silva from the University of Colombo, Department of Zoology and Environment Sciences. I will be presenting to you about my work on the polymorphism of dopamine receptor D4 gene in horse swallows from mountain human settlements. Personality traits and types are not limited to humans, but are also found in the animal kingdom, more notably in mammals and birds, identifying them as unique individuals. Personality can be defined as repeatable behaviors that vary between individuals but remains constant within an individual over time. It is a complex trait, meaning that it has both an intrinsic component embedded in the genome of an animal, as well as it is influenced and shaped by external factors, such as the environment an animal develops in. It is said that 20-50% of animal personality traits happens to take, and a given personality trait is not merely the work of a single gene, but the result of several genes interacting as well, which is identified as homeostasis. Also, differences in personality traits among individuals arise due to genetic polymorphisms. The final outcome would be the interaction between the genetic component and the environmental factors. It is DR4 gene is one such gene that has been linked to a variety of personality traits. It codes for a type 2 dopamine receptor, namely the dopamine receptor E4. Its ligand is the neurotransmitter dopamine. This is a G protein coupled receptor and is part of the dopaminergic system of the brain that acts as a rewarding system. When bound to dopamine, it induces an inhibitory reaction. Personality traits that is known to be controlled by DRD4 are nervous taking, early exploratory behavior, risk taking, wariness and boldness. So roughly speaking, these traits are important when adapting to new settings and exploring things. That being said, we are experiencing unprecedented rate of environment and habitat change presently in the present day due to human activities. We see it as urbanization and human constructions. And so are the animals in the wild experiencing these changes as well. The habitats are being converted into human settlements, agricultural lands, and industrial areas, leading to a drastic change in habitat structure and resource distribution. Moreover, the penetration of humans have been introduced many novelties to them as well. And in order to survive, wild animals should be capable of exploring the new environment to find food and nesting sites. They must also have a chance to the presence of humans who would be viewed as a potential threat. So how do they adapt to these shift of basic ecological paradigms? In order to understand this, I use house fallow as a model of the risk. They are found in both distant natural habitats as well as in human settlements. Right here, nesting on man-made structures, thus have been able to make the transition from forest to the town successfully. Is there a change in the genetic structure with respect to polymorphism of DRD4 gene? That was my question. That was shaped by human activities that ultimately resulted in changes in their behavior and personality. And I hypothesize that correlation exists between the level of DRD4 gene polymorphism and the degree of urbanization. And this degree of urbanization was taken as a proxy to the amount of human activity in that area. The main objective of the study was to explore how genetic diversity and DRD4 varies with the degree of human activity. These kind of studies are important for us to understand how our activities impact wild animals and their behaviors and personalities at a molecular level. Moving on to the methods of the study, I conducted a field scouting session to locate these swallows and their nesting. So I used research to capture the birds and obtain blood samples. 51 pieces of blood from the reception of the birds were obtained and stored in the laboratory at the minus 20 degrees Celsius until used for further purposes. So these samples from several locations in Sri Lanka, namely Holland, Amazon, Ohio, Kotelawala, 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 and Kotelawala. Samples were obtained from Western Malaysia and Borneo as well for genetic analysis. At the laboratory, we extracted DNA using a Kiachen. I extracted DNA using a Kiachen, DNA using blood and tissue case. Two regions of the DRD4 gene were suspected to have polymorphisms were amplified using PCR. These two regions were the first zone of DRD4 and the third zone of DRD4. These were the primers I used for identification. And the resulting PCR products were gel purified using Kia picture extraction kit, and there was a sequence in both the forward and reverse direction. I assembled and aligned sequences using the Genius software as well as the Mega 10 software based on the requirement, and I obtained the values and statistics was calculated using DNSP version 6. An urbanization gradient was prepared using a scoring system. Using several features of the landscape, and these maps and the landscape features were drawn using Google Earth. And these were the features I used. Those were human construction, agricultural plots, roads, forest cover, water bodies, and other undisturbed natural habitats. Moving on to the results and discussions, this is what my urbanization gradient looks like. According to the graph, the lowest levels of human activity exist in Northern Plains, Ambedkar, and Fairfax. Highest levels exist in Kotelawala and Kotelawala. I found that the third axon was variable between individuals in Kenya. It was polymorphic between individuals, but the first axon was not. It was identical among all individuals. This is how the variation of genetic diversity parameters, namely the nucleotide diversity and the heterozygosity, are varied along the urbanization gradient. And according to my results, human had an eroding effect on the genetic diversity of DRD4 exon 3. The graph shows a clear decline of both heterozygosity and nucleotide diversity when moving less urbanized to high urbanized areas. And since DRD4 is responsible in generating personality traits involving adapting to novel environments and overcoming disturbances, we can say maybe the genetic diversity dropped because some genotypes were filtered, and that those found in the urban areas are the ones beneficial for the survival of these birds. But in the national habitat, more variation in personality exists, which might explain the high level of genetic diversity found in those areas. And I also calculated the touchy statistics to observe if there have been any demographic events in the population of twin swallows in Sri Lanka. However, the non-significant values indicate that no demographic processes such as population expansion or purifying selection has taken place. In conclusion, the nucleotide diversity exon 3 of DRD4 in house swallows has a correlation with the degree of urbanization of human activity, nucleotide diversity and heterozygosity at the third exon declines along the gradient of human activity. And we can say by looking at that the genetic diversity in DRD4 gene of house swallows are affected by human activity, meaning that habitat alteration and landscape change by man affects the diversity within populations with respect to personality traits associated genes at a molecular level. This also implies the genetic basis for habituation and adaptation in wild animals. So, this concludes my presentation. These are my key references. And I acknowledge my supervisors, lab mates, and the funding organizations for their support. Thank you.
Thank you, Mr. Isidua, for your nice presentation. You are having lots of confidence in what you do and what you believe in. That is very good thing. Yeah, I mean it. Yeah. Anyway, the presentation is open for discussion now.
Okay.
Yeah, I have a couple of some curiosity in me. Why hill swallows? Are there any other bird species which are better? I don't know. Please tell us.
These kinds of studies have been conducted in other species in other countries as well. I selected the hill swallow because the hill swallow has a very striking difference in their selection of habitats, because they also exist in natural areas and they also exist in man-made human constructed areas. So I have in my presentation I have shown that they exist in both types of locations, and it's very interesting to observe that they are able to thrive in both types of landscapes, in changed landscapes as well as in natural landscapes. So there has to be some difference in their personality or behavior that enables them to do this. That is why I selected this species to study this phenomenon. And using a bird is way more easier than using mammals. That was the reason I used a bird model. So this is an ideal candidate organism for me to explore my research question. That is why I used the hill swallow.
Yeah. What is the singular name of this hill swallow?
It's known as the Kandu ahirihinia, sir.
Kandu? Could you repeat?
Kandu. Yeah, yes.
Yeah. Yeah. Yes, you have amplified an area spanning intron 1 and exon 3. I mean, is it one amplicon or two amplicons in your study? Have you amplified them separately? Have you amplified them separately or together?
Separately. I performed PCR on intron 1 and exon 3 separately as two separate reaction cells.
Yeah, I'm wondering why you amplified the intron in a study which targets the polymorphism and genetic diversity, because we know that introns can accumulate changes very, very freely without any influence from any selection pressures, because selection pressures hardly apply on introns. Can you clarify it, please?
Yes, I am. Only because in a previous study, they have also amplified the intron, the first intron, and they have suspected there could be some variation, some form of polymorphism with respect to changing of the splicing sites, such variations as that can be found in an intron. That is why I amplified the first intron. It was one of the suspected reasons to have a polymorphism with respect to this. So differences in splicing sites can also create different proteins, different receptors, which can result in a different kind of activity in the brain. So this is a dopamine receptor found in the brain. So depending on the configuration of the protein, the way dopamine binds can vary, and it might have pleiotropic effects based on polymorphisms found in the first intron. That is why I amplified that to see if there are any variations with respect to that.
Yeah, good. One more thing, construct as a statement.
Yes.
Yeah, your concluding remark reads that habituation to human instruments has an eroding effect on genetic diversity in a personally related gene in cells. Actually, when there is a change under an environmental force, we can't just say it is eroding or constructive in evolution. It is just a change. So that is the change probably that species need to survive under the new conditions. So using of modifiers that has to be done under lots of consideration. I hope you already are aware of these things. One more small thing is in the top area of your abstract. Insulos are aerial insectivores confined to mountains of Sri Lanka, and birds are not aerial. They are territorial. We don't have aerial animals.
Because they are found mostly flying. They very rarely restaurants and trees and such. That they are categorized and sort of identified as area. I know it's kind of weird to call a bird area.
Yeah, you know your story, right? Right. It's a very good effort, Ms. Silva. Thank you so much for submitting this paper to the research, international research conference of Kotelawala University. And thank you so much for your authors and everybody involved. Thank you.
Thank you very much.
Oh, there is another question, please.
One question. What about the ethical issues related to this research? Have taken ethical clearance and
Yes, we obtained permits from the Department of Wildlife before conducting the field, before the field sessions. That those are the types of regulations that are required in our country before doing such field sampling. So it was only a permit.
it is same as the ethical clearance from you are taking from your university or some other ethical committee. Same.
Yes. Usually, when we are obtaining samples from birds and other wild animals, we have to take permits from the Department of Wildlife. So we did. Yeah.
So that is not ethical clearance, no? So no need to take ethical clearance from the relevant ethical committee. I think that wildlife that permission
Yeah.
is not same as the ethical clearance.
For studies like this, we don't use the animals other than obtaining samples and morphometric measurements. So usually, as I am aware, ethical clearance committees separately are usually obtained for human studies. This required only, since this is a wildlife-related experiment, this required only a permit from the Department of Wildlife.
So, but in our case, we have to take the ethical clearance even we are working with mice models and that kind of animals.
Yeah, with mines, I'm also aware that you have to obtain ethical clearance. But in this kind of experiments, it's usually only the Department of Wildlife permits that are being obtained.
Actually, ethical clearance is possible. You can try obtain ethical clearance for this kind of studies from Institute of Biology. Yes.
And the thing is, first we have to take the ethical clearance before starting the research.
Yes, yes, I'm aware.
Yeah, actually, animal welfare is an increasing concern in modern world, and so is
Yes.
and scientific committees even more focused on the issues related to handling of animals and their ethical nature. So it is better to obtain ethical clearance from a relevant authority or organization before venturing into any research nowadays.
Yes, usually, so if I may mention, in phylogenetic studies and such, it's usually the wildlife permit. So this was also kind of similar to one of those studies with respect to handling and sampling. And within the wildlife department, they do undergo a sort of wild animal welfare review, I guess. Before giving us the permission.
Yeah. Think about it.
Yes, I will. Thank you very much for that comment, sir.
Yeah, sure. You are welcome. And are there any questions? No? Right, once again, thank you, Ms. T. Silva, for participation in this technical session.
Thank you very much, sir, for having me.
And yeah, that concludes our presentations of our technical session. And I have to say, the three studies we presented were very refreshing and encouraging for even aspiring students of any audience. So take them as examples. We are a country that needs progressing in many areas that include science and technology. Knowledge is the essential substance, essential resource which drives any future progress in any country. So no knowledge, no progress. So therefore, the people who are involved in generating new knowledge, that is researchers, are doing a very noble effort on behalf of all the countrymen and humanity as a whole. So we are about to conclude the session and we are now stepping into the concluding items.